Welcome students. So today we would be discussing about mucosal immune system. That what is mucosal immune system? What are their types? What kind of cells and organs are basically taking part in this mucosal immune system? And you will be getting to know the complete understanding of different mucosal surfaces and the cells and organs that play a role in providing mucosal immunity. So first of all, you need to understand the term immunity, that what is immunity. Basically, the immunity is our body's capability to resist or eliminate the potentially harmful foreign material. They can be infectious microorganisms, they can be toxic products, it can be anything, or there can be many abnormal cells. For example, if our body cells, they become cancerous. In that case, also our body immune system come into picture and they, start eliminating that thing. So this can be many things. So immunity can be provided against a range of things, for example, virus, carcinogens, pollution, dust particles, germs, parasites, bacteria, fungi, toxins. So all of these entities are basically resisted by our inborn immune mechanisms. Now the point is, what is immunology? Now, having talked about what is immunity, next point is immunology is a branch of science, or you can say it is the study which basically involves the role of all type of immune cells in providing the protection to the host body from foreign macromolecules or invading microorganisms, and the kind of responses that are triggered in response to those kind of foreign entities. Now, who is the host? Host can be, for example, I am the host, I am a human. If I got some infection, in that case, I am the host, and that infectious entity is the foreign material. So, the foreign macromolecule or a foreign antigen, it can be a bacteria, it can be a virus, it can be some proteins released by them, it can be some toxic materials released by them, it can be parasites. So, everything that is not a part of my body, that would be considered as a foreign material or foreign antigen. That means immunology, this branch of science is basically is arising from a basic science, and the major clinical applications are it provide it basically plays a very important role in providing post defense reactions that are normally generated in response to some foreign antigen or substances which are not considered by our body as self material. So all of these things basically provide the host defense mechanism a quite strong foundation that they can fight against any kind of infection. Now, here are the few definitions that, uh, however, we have discussed about immunity, immunology. Now, the point is, what is immune system? Now, our immune system, it basically comprises of all cells, tissues, and other molecules that will basically play a role in providing or in mediating resistance to the infections. Immunology, as we have just discussed, study of structure and function of various immune system. Immunity is the resistance of a host towards pathogenic microorganisms and toxic effects. And immune response, now the point is, what is immune response? Immune response is actually a quite a coordinated and collective response that comes towards any infectious entity by the different immune cells, tissues, and organs of our body. You can say a coordinated response to the introduction of foreign substances inside a host body that is basically mediated by many cells and molecules of the immune system. So the main role of immune system of a host body is to provide defense against microbes, provide defense against the growth of tumor cells, for example, cancerous cells, so as to block the growth of those cells at that very position, and the regulation of homeostasis. That means whenever the cells are, if they have developed abnormally or there are some dead cells, in that case, those cells should be cleared out from the body. In that case, immune system is the major, the key player who, activate different cells and tissues in response to that. Now, if we talk about that, how a particular, human body can be invaded. So, and how we get protected? In that case, if you think about it, the main anatomical barrier in providing immunity to any kind of infections or any kind of injury, that is our skin, right? So the skin is exposed to the external environment. That is the major exposed organ is skin. But this complete skin is completely sealed. It, however, it is comprised of three layers: dermis, epidermis, and all of these things they contain many substances. They are being produced by the skin. However, now the point is, what is mucosa? Mucosa is basically it is the extension of skin because it will line our different digestive tract starting from the mouth to the anus, and the total surface area is around 200 times greater than the skin surface because it is providing a complete lining surface to all the internal lines. So, what are mucosal tissues of the human body? So, if you talk about these are the like primal glands, they also have a mucous lining, salivary glands, mammary glands, kidney, uterus, bladder, complete urogenital tract. On the other hand, conjunctiva, oral cavity, esophagus, stomach, complete respiratory tract. So, there are different type of all organs are completely lined by these mucosal tissues. Now, the point is what is mucosal immune response and what is systemic immune response, because we are talking about mucosal immune system. So the major isotype, the major difference is whenever you are talking about a mucosal immunity, in that case the major antibody that is produced is dimeric secretory IgA, because this is the only antibody that is released in the secretion. For example, in the breast milk or in the various kind of mucosal secretions inside the intestine. So most of the antibody producing cells and effector T cells, they are normally localized in a tissue that is known as MALT, that is mucosal associated lymphoid tissue, because these tissues they have the capability to to produce these secretory immunoglobulins also. However, they are completely distinct from inductive and effector lymphoid cells. If you talk about our you must have heard about it that there are many bacteria that are colonizing our intestine. So those enteric microflora, that enteric bacterial flora, that basically act as a stimulus for the development of mucosal immune system so as to protect the mucosal surfaces from any kind of infection. Now there are two type of mucosal surfaces, type 1 and type 2. If you talk about type 1 mucosal surfaces, then type 1 mucosal surfaces are generally covered by the simple epithelium. That means they normally express a very simple polymeric IgG receptor that allow the secretory IgA to enter inside the lymph. And the major sites are intestine, lungs, and uterus. These are type 1 mucosal surfaces. Now the point is, what are type 2? What is the difference? Type 2 mucosal surfaces are basically covered by stratified squamous epithelium, as you can see here. These are stratified squamous epithelium so as to provide protective barriers for the activities that are important for those species. And the major sites are our oral cavity and vaginal cavity. Now the point is, having talked about what is mucosal surface, what is mucosal immunity, what is the basic immune system, immune response, immunology. Now the point is, what are the cells of these immune system, these mucosal immune system. So, depending upon the cells and organs, cells they are divided into two types. Innate response. In case of innate response, there are many cell types that play an important role. However, in case of adaptive or specific immune response, lymphocytes are the major key players. And if you talk about organs, they are of two types: primary and secondary. Primary organs are those where lymphocytes they basically develop and they get matured, and the secondary are those which receive mature lymphocytes from primary lymphoid organ, and they interact with the antigen at that very site so as to mount an immune response against a particular foreign entity. Now if you talk about our circulatory system, that is blood, and if you talk about our lymphatic system, the main fluid is lymph. Now if you talk about cells, Cells means leukocytes. Leukocytes means white blood cells. So they are divided into two types: granulocyte, non-granulocyte. We also call it as agranulocytes also. So in category of granulocytes, there are three types: neutrophils, eosinophils, and basophils. And agranulocytes are monocytes and lymphocytes. So if you talk about, if you normally conduct an experiment in the lab by taking a blood and you allow it to, if you want to separate different type of cells, then we use Fecal reagent. In that case, if you centrifuge the cells inside the reagent, the leukocytes would be forming a buff-colored layer, buff coat, and because the rest of the content, because maximum amount in our blood is plasma, that is around 56%. It is fluid part. 1% is RBC. And if you want to, basically have a different kind of scenario, you want to isolate plasma and serum, then plasma is which is containing an anticoagulant molecule and serum is after coagulation. That means if you want to have a serum, in that case you allow the blood to clot and the upper liquid is known as serum. So these are the different things. Now the point is from where these cells are coming, how they are synthesized, what are their functions. Now the main cells of these immune system, they arise from a pluripotent hematopoietic stem cell, that is HSC, through two major lines of differentiation, that is myeloid and lymphoid. Myeloid lineage. Myeloid lineage cells are basically involved in producing phagocytes, for example, neutrophils and other type of cells. However, if you talk about lymphoid lineage, they will produce only lymphocytes. Now, what is hematopoiesis? Because the process of formation of these cells, that process is known as hematopoiesis, and the starting cell is pluripotent hematopoietic stem cell that give rise to second generation stem cell with restricted lineage potential. That is why we call it as different progenitors. So this is the complete differentiation pathway. As you can see, this is the hematopoietic cell. It is self-renewing. Now that there are two lineages, myeloid and lymphoid. From the lymphoid lineage, natural killer cell is being formed, T cell and B cell. That can be further differentiated into T helper and cytotoxic. B cell will be differentiating into plasma cell producing antibodies and dendritic cells. Now, in case of myeloid progenitor, the main cell types are it can either proliferate into dendritic cell or it will result in the formation of granulocyte monocyte progenitor that will give rise to neutrophil, then eosinophil, basophil, platelet, erythrocyte. All of these things are basically originated from a myeloid progenitor that we must understand that how these cells basically develop. Now, whenever some kind of infection is there, in that case, what are the first responders? If you talk about this thing, if you see, this is the hematopoietic stem cell. This is a common myeloid progenitor because these cells are the first responder towards any kind of infection. As you can see, this is the granulocyte lineage that is being developed in which eosinophil, neutrophil, and basophil will play a role because basophil will release histamine, neutrophil will perform phagocytic action, eosinophil will have phagocytic action towards parasitic infections if it is happening. So these are the first responders, or you can say they are the first kind of immune cells that get activated in response to any kind of uh, infectious attack. Now, granulocyte, that is the first, or you can say front line of attack during an immune response. The main differentiating feature is the presence of granules inside the cytoplasm. And these granules, they are basically responsible for mounting an response because these granules are released only when they interact, when this cell will interact with some pathogen. Then these granules will release proteins or toxic materials with distinct functions. Either they will kill the target cell, they will regulate other T cells and tissues or signal them so as to kill those or eliminate those target cells. However, all of these granulocytes they have found to be having multi-nucleus, of which neutrophil. This is the first and the main effector cell in the innate immune system, and 50 to 70% of our blood cell it is basically comprised of these neutrophils. and they are basically released from bone marrow, and they have the capability to circulate. Basically, they are surveillance agents of our immune system, because they keep on circulating in the body and keep on checking whether some infection is there or not. If they happen to found some kind of infection, they will directly attack on it so as to clear it at the early stage. So they normally keep on circulating for around 7 to 10 hours. They enter tissues, they can cross all the barriers, and they live. However, the lifespan is quite short because they live only for few days. After that, they again, get recycled. So the number and recruitment increase. That means the number of these neutrophils and the recruitment strategy to a particular tissue, that normally is exponentially increasing in case of certain kind of infection. That process is known as mucositosis. This is the major diagnostic procedure also. The neutrophil count is quite high. That means your body is basically responding. However, this is also the main cellular component of pus also. Whenever you are having some tissue injury, at that time a white color pus is formed. That pus is basically due to the excessive accumulation of neutrophils at that very site to fight against the particular infectious agent. Basically, why we have named it as neutrophil, because it is named based upon the staining qualities of the granules. It is having a multi-load nucleus. It is polymorphonuclear. That is why we sometimes also call it as PMN. This The granules of neutrophils, they normally stain light blue to pink color. They can circulate around 7 to 10 hours. They migrate into the tissues. They are the first responders. They are motile in nature. They are phagocytic in nature. They express, if the count is quite high or they are showing the symptom of leukocytosis, that means some infection is there. They basically act as an extracellular trap. That means they first of all respond to the extracellular infections. Now, how these neutrophils basically shape the immune response, how this cell basically provide us the immunity. These are the immune modulating mechanism. This is the endothelial cell, and you can see the main mediator is granule protein. It can be different ones. There can be many chemokines and cytokines playing a role. There can be direct contact between the receptors. There can be, if you talk about endothelial cells, these are the main things. Target is endothelial cells, because all of these antimicrobial mechanisms that are being imposed by this neutrophil, it is divided into four parts depending upon the cell type. As you can see, these neutrophils, they have antimicrobial mechanism. Why? Because they can work as a phagocytic cell, so they will trigger the process of phagocytosis. They can release antimicrobial peptides, and they can release net. Now the first point is, if they are attacking endothelial cells, then the granule protein, chemokines, cytokines, direct contact can be the major key there. In that case, endothelial cells get activated and vascular permeability is increased so as to clear that very infection. Now, in case of monocyte macrophage, many granule proteins, chemokines, and cytokines, however, the number and the count would be different. They basically get recruited, they impose antimicrobial activity, they secrete cytokines, they secrete chemokines because their count is increased. On the other hand, if dendritic cells are there, in that case, this neutrophil, they will, they will be releasing certain granule proteins, they will act as an alarming agent to these dendritic cells, so that these antigen presenting cell can reach to the site and they can capture the antigen, process it. So they basically get recruited, they get matured, they secrete cytokines, they secrete chemokines. On the other hand, the last one is, sometimes APC-like neutrophils are also playing an important role, in which soluble mediators or soluble proteins are being released. In that case, T cell proliferation, suppression, recruitment, activation of T helper subtype, and the clonal expansion is basically triggered. This is the next, that is neutrophil express trapping system. This is a neutrophil testing, that how they get, after activating, they trap the infectious moiety. That is why we call it as net. Another cell type is basophil. However, the count is very less, because in our blood cell, the least counted cell is this one, that is around less than 1%. They are non-phagocytic. They do not follow the process of phagocytosis. However, its nucleus is white and all, the granules are stained blue in color. They sometimes, play a role in some allergic reactions. Whenever some allergy is there or some hypersensitivity reaction is being triggered, in that case, this basophil count will increase abruptly. And critical response comes if some parasitic infection is there, because the main role is They will bind the circulating antibodies and they will trigger the release of a molecule known as histamine. And that histamine is basically responsible for increasing vascular permeability of blood vessels. These are these mast cells that are normally, after staining looks like this, and they will release histamine. Another cell type is eosinophil. This cell is having a bilobed nucleus, or you can say bean-shaped nucleus. It is quite motile. Sometimes it is phagocytic against some parasitic infection because they will kill antibody coated parasites by binding to those antibodies to the FC by the help of FC receptor. And the degranulation mechanism basically comes into picture so as to, they release certain kind of substances so as to kill the parasite. These are the few proteins that are present in the neutrophil, eosinophil, and basophil that we have just discussed. In case of neutrophils, proteases are there, antimicrobial proteins are there, protease inhibitors, histamine is there. Examples are elastase, collagenase, defensins, lysozyme, antitrypsin. Basically, they act in clearing the pathogens, and vasodilation and inflammation is a major role. Eosinophil cationic proteins, cytokines, chemokines, ribonucleases, basophil, cytokines, histamines, lipid mediators. And all of these are basically showing different kind of response by bringing about the vasodilation, initiating the inflammatory response, smooth muscle contraction, antiviral activity, attracting the leukocyte to the site of infection. Now, the point is this myeloid antigen presenting cell, that means monocyte, macrophages, and dendritic cells from which they are arising, they are phagocytic in nature because the main function is ingestion of foreign molecule, digestion into the peptide, and then they present those peptides onto the cell surface. Basically, they act as a bridging cell between innate and adaptive immune system because they make contact with the antigens at the peripheral region and they interact with the lymphocytes in the lymph nodes so as to get activated. And they secrete certain kind of proteins so that all the other immune cells they get activated and reach to the cell. So this is the monocyte role. Pluripotent stem cell in case of myeloid progenitor, the monocyte will and macrophages will get activated so as to induce a phagocytic function. Now, monocytes, they are mononuclear in nature, they circulate in the blood, and this kind of staining is happening. Now, the point is what are macrophages? Those monocytes who enter into the tissues, when these monocytes they cross the barrier, they enter inside the tissue, they get mature in the form of macrophages or dendritic cells. They get enlarged, they have some phagocytic function. However, they can be free or they can be tissue specific because there are certain type of macrophages, they are localized in particular kind of cells. For example, Kupffer cells of liver, in that case the name becomes different. The main function remains the same because they will digest the antigen, they will present the antigen because they also express certain kind of receptors onto the surface. As you can see, the process will remain same. Phagosome formation, these are the pseudopodia which is basically engulfing the material. Phagolysosome would be formed and then complete killing would be happening. Now, what are dendritic cells? These are heterogeneous myeloid and lymphoid origins because they get originated from both the progenitor lines. And they are the best antigen presenting cells for presenting naive T cells. First of all, it was discovered in the year 1970, for which a scientist also received the Nobel Prize in 2011. And they played a major critical role in mounting immune response against any kind of infection because they can capture antigen at one place. Suppose they have captured antigen at a tissue injury site, then they migrate to the lymph node so as to present it. That means they are capturing the antigen from one place, they migrate with the captured antigen to another place. So they basically experience the phases of immature to mature, and because of this complete capturing and presentation, the functionality also changes. These are the dendritic cells under the microscope. They will look like this. These are the few stained pictures. Sometimes they give a appearance of flower if you go for stem. So when these myeloid progenitor, they also how they get differentiated into platelets also because these platelets also play a major role because in certain kind of diseases the count of platelets also matters. These are basically they are originated from megakaryocytes. These megakaryocytes give rise to large number of platelets. So this is the kind of picture that we normally observe under microscope. If some blood clot is there, in that case, these yellow threads that you can see, these are the fibrin threads that basically resulted from to the activation of fibrinogen. This is the blood clot. Now the another point is how the erythrocytes are being, migrated. In case of parasitic infection, the erythroid progenitor give rise to erythrocyte also, so that they can migrate into tissue. Many of the mature human and mouse RBCs, they do not express some kind of nucleus. In that case, the RBCs sometimes impose different kind of appearance. Now, when the adaptive immune response is mounted, in that case, lymphocyte, lymphoid lineage get activated, natural killer cell, B lymphocyte and T lymphocytes are produced and they, basically get activated. So we'll be discussing these cells of mucosal immune system that are lymphocytes. Lymphocytes, there are three types and it mainly comprises of 20 to 40% of WBC. However, you cannot distinguish them morphologically. You basically distinguish them depending upon their function. For example, T cells, they are divided into two types, two main types actually. Helper CD4 positive cell and cytotoxic CD8 positive cell. Helper T cell, they basically recognize antigen in conjunction with MHC2 and cytotoxic with MHC1. In case of B cell, they get differentiated into plasma cells so as to secrete the antibody. And natural killer cells, they are the main part of innate immune response also. However, they act as a bridge. So these are the T and B lymphocytes. They have a large nucleus with dense heterochromatin, and there is a very thin ring of cytoplasm, as you can see. This is the complete nucleus part, and this thin ring, this thin rim of cytoplasm is there. And the major difference is they play an important role in recognizing specific antigenic determinant. That is why the main feature of adaptive response, that is specificity and memory, that is basically triggered by these lymphocytes. These are the examples that we have just discussed. Lymphocyte, these are the T helper cells, cytotoxic T cells, these are the B cells. These B cells get differentiated into plasma cells, natural killer cells, and these are the natural killer differentiation. And this is the complete cascade. This is the common CLPs, common lymphoid progenitor. It is differentiating into three types, NK, B, and T. T would be differentiating into two types, that is T helper and cytotoxic. And after that, this T helper cell have different subtypes also. For example, T helper 1, T regulatory cell, T helper 17, T helper 2. This T helper 17 is basically, produced in response to some fungal infection so that it can release interleukin 17. Now, these are the few markers depending upon which we can differentiate these different cell types. As you can say, B cell only express CD21 or complement receptor 2. And along with that, FC receptor, complement receptor 1. There are two co-receptors that are involved in the signal transduction, CD40 and CD45. However, in case of T helper cell, they express CD2, CD3, CD4, and what you say, CD8. There is a co-stimulatory signal CD28 that binds with the B7 molecule. This is CD45 that acts as a co-receptor. In case of cytotoxic T cell, there are also the same kind of cells. In case of natural killer cell, the molecules or the differentiation pattern of the CD markers basically differs because it expresses CD2, CD5, CD8, CD16, FC gamma receptor 3, and lastly CD45 and CD56. This is the condensed heterochromatin. You can say a T or B lymphocyte in the resting stage. At that time, the diameter is 6 micrometer. But once they get activated, this complete heterochromatin, they will get that will get decondensed and cytoplasmic ring would be enlarged. In that case, the diameter will increase to 15 micrometers in diameter. This is in the case of plasma cell, that is in the case of B cell, that normally lives around 1 to 2 weeks, and they have the capability of secreting 100 to 1000 of antibodies molecules per second. And again, the size is around 15 micrometer in diameter. So all of these cells, plasma cells, and there are large amount of Golgi and abundant layer of endoplasmic reticulum that play an important role. These are the three kinds of antigen presenting cells. That means how they basically act. These are the dendritic cells that they present antigen to the T cells so that they get activated. They capture the antigen, process the and present the antigen. These are the dendritic cells. Under the microscope, they will look like this. This is the picture, and sometimes the same photograph is something like this. In case of macrophages, they appear like this, and in case of SEM photograph, it is something like this. These are the B cells producing antibodies. This is the main condensed heterochromatin with thin cytoplasmic ring, and after that it will get decondensed, and in the SEM photograph will appear something like this. So these are the few MCQs pertaining to this lecture, that which kind of cells are playing important role, what kind of chemicals are being released, what are the major differences, which how you can differentiate different cell types from one another depending upon their structure, depending upon their function, depending upon their circulation ability, how they are being originated from a particular progenitor cells, for example, whether lymphoid or myeloid, how they are basically attacking or providing the comprising the mucosal immune system, what kind of antibodies being released in the mucosal secretions, how they are originating, what are the different type of mucosal surfaces. Now in the next lecture we would be completely covering our organs of the mucosal immune system, and how they are basically playing the role in providing the mucosal immunity. Thank you so much.